part 2. Hey guys, this is part 2 of my video for the bio benchmark. Okay, our next concept is proteins can do gene regulation. Okay, this basically means they can turn genes on and off on the chromosomes. Okay, so gene regulation basically means controlling the sets of genes. Anything from DNA replication all the way to them compacting into chromosomes for DNA replication. Protein does the whole ordeal. Without proteins, you wouldn't exist. For the fact that the cell exists is only sustained upon the protein itself. Now, they can turn on and off the genes of the they can turn on and off gene regulation. Okay, this process is known as transcriptional regulation. Okay, proteins can also regulate gene expressions at other stages such as RNA processing, RNA transport, and translation. Okay, so an example of this can be transcription factors. They are proteins that bind to DNA and control the rate of transcription, or the rate of them being translated to RNA, right? A very famous example is histones, okay? Histones, this is an example, you don't need to memorize this, okay? There are proteins that package DNA into a compact structure called chromatin. And if you already know what chromatin is, it's the one bar of a chromosome. Okay, these proteins all just help with regulating. Okay, there's a lot more ordeals when it comes to proteins, but I think the general idea and the general gist is that they can do gene regulation by turning on and off the gene chromosome, and gene regulation in the fact that they help in transcriptional regulation. Remember, proteins vary, like topoisomers are so important for DNA replication. That's a part of gene regulation. What is it? The scissoring one. You know, watch my DNA replication video for that. tRNA, right? They have proteins in tRNA that help, right? Those also help in gene regulation. In fact, tRNA itself is not even DNA. It's a protein. tRNA, right? You got to understand these things. Okay, proteins help in so many factors. Okay, remember one thing: if it ends in ace, eons like histones, right? Oh, I spelled histones wrong. My bad, gang. I spelled histones wrong. It's histones. That's an N, by the way, right? If it ends in ones, if it ends in ace, right? You know those are automatically proteins. Okay. So that's a general concept here. I don't want to delve into it. If you think this is an important topic, right, go ask my source for it or call me in VC. I'll go a little bit more detail into it. But we have a lot more things to focus on, so I physically cannot go any further than I am right now. Okay. Our next topic, guys, is define non-disjunction. Disjunction. Good Lord. I don't even understand these crap. Okay. Non-disjunction is basically failure. Okay, let's talk about gene regulation, right? So I'm not going to go so detailed into it because we're going to go to extreme detail later on. Okay, that's going to be in our next video. That detail will be unmatched. But this one is just the definition of non-disjunction. Non-disjunction, right? Non-disjunction. Okay, what that basically is. Let's say this is a cell, right? I'm going to favorite this and favorite this, all right? This is the mother cell. This is the dad cell. What should typically happen, these are chromosomes, by the way. Chromosomes. What should typically happen is that the cell should be split into two, right? And one-on-one, right? Dad gets its own cell. This is the concept of homologous chromosomes. So this happens during meiosis. Oh, I need to note that down so bad. Happens during meiosis, right? Right? So what should happen is that the dad cell gets its own cell and the mom cell gets its own cell. And then what should happen next after that, I think you guys can already tell, all right, is that the dad gets one of their chromatins and one of their chromatins, which then duplicate, and then it starts mitosis process. We'll get into that later. And then the mom cell would take their one chromatin and become one chromatin. That's how it should normally go. This is how meiosis should work. Now, non-disjunction, right? Pertaining to non-disjunction, all of the processes could be the same, right? From step 1, this is perfect. Step 2, this is perfect. But then when we reach row 2, maybe the dad chromosomes are perfectly fine. But the mom's, or dad, I don't know, whatever gender it is, right? Both of the chromosomes could be 1, and 1 could be failed, right? And it receives nothing, okay? That's the form of non-disjunction, where it's technically an issue, okay? Non-disjunction is when a mistake happens during meiosis that leads to an irregular amount of chromosomes in one gamete, or gametes as in these cells, right? When there's not a normal amount, you can easily tell that there's something wrong. And this happens during the splitting of the cell, okay? To put into detail, when the centrioles comes in, or centrioles, right? When they come in and they take each of the chromosomes, right? Maybe one of the centrioles take both pairs of chromosomes, and this one just has nothing. And that causes one of the cells to just not have anything in it. That's what non-disjunction is, when there's an issue during DNA replication. All right? I hope this is the easiest understanding for it. We're going to continue on. Any questions, just text me on Discord. I'm not going to make a whole, go into so deep detail into it. The next thing is define karyotype. What is karyotype? All right? Whoa, my bad. All right. Here's how I'm going to explain this. Okay. Have you ever seen an image like this? Karyotype human. This is very famous. You can search this up in the National Genome of Research. You might be seeing this in a lot of textbooks. And as you continue on with genetics, you'll definitely see this often. This is a karyotype. Not the gene specifically, but how it's arranged. This is a photographic, or a karyotype. Karyotype. Photographic arrangement of a full set of chromosomes in a cell or organism. What this basically means is that the way that you're going to set up those chromosomes to make it into a photographic arrangement for a person to understand this organism's genome or their chromosomes is a karyotype. The way that these chromosomes are set up like this is an example of a karyotype, where they're all just a photographic arrangement of the chromosomes. All it is that the chromosomes are arranged in such a way and then photographed in a specific way, and that's what a karyotype is. This should not be a difficult topic to understand, and you definitely do not need my help for it. Okay, you should understand this is just a photographic arrangement. Okay, our next topic is define, selective permeability. Okay, this one's going to be a hoodie. I know this one like the back of my hand, but I need you guys to know this at the back of your head as well. Okay, we're going to make a phospholipid bilayer. What is a phospholipid bilayer? That's a cell membrane. Phospholipid bilayer. Okay, we're going to draw one out real quick. This is our phospholipid bilayer. Sorry if it looks whack, but let's try and get it. Okay, what are phospholipid bilayers? I need you guys to know this. You guys should know what a phospholipid bilayer is. And if you guys don't, let me help you right now. Right? Phospholipid bilayer is a lipid. It's in the name. It's a fat. Right? It's a fat that looks something like this. Right? Where the outside loves water. Just put a heart. And this one hates water. The tails hate water. So what it does is it positions themselves so that the tails can go inward, so that basically little to no water comes in, and the heads go outward so they can constantly touch water. It's a special kind of lipid where it doesn't just fully hates water, but it likes both. Now, what this allows it to be selectively permeable. What does selectively permeable mean? Selectively permeable. Okay, this means it allows some things in, and it doesn't allow some things. Allows some. It's allows some. That's literally the meaning. It allows some things in, and it doesn't allow some things at all. Now, what does it allow in, and what does it allow out? Now, we're talking about the concept specifically to cell membranes. Okay, remember how the inwards is a lipid. Let's say a molecule like for goodness sake, glucose, right? Let's say a big molecule of glucose comes in. Now, this is definitely not the shape of glucose. It easily can pass through the water-loving state because glucose is easily loving with water. But when it enters that lipid state, it has some struggle, right? And that what makes it selectively permeable. The phospholipid bilayer allows some things to come in and some things to not. But what does it allow to go in and go out? It's things that are fats, right? The only thing that these tails like is fats themselves. They hate water. And what's the opposite of water? Fats, okay? So the only thing that are going to allow is fats to go through. So fats like cholesterol, right? Fats such as omega-3 fatty acids that are found in fish easily can go through a cell membrane, right? These fats can easily just go through and can easily pass through the small intersections of the water-loving state and go straight through. But it's not just those. It's just really, really small molecules as well. Something as small as OO, which are just two tiny oxygen atoms, can easily go through the membrane even if it's not water-hating, just like the tails. Since it's so small, it just passes through. Right? That's what permeability means. That some things can go inside the cell membrane and some things cannot. Okay? So that's a general sense of cell membranes. Alright? Selective permeability, some things go in, some things don't. Now we're going to go to the types of diffusion. This is a very, very, very long lesson. So, get ready for this one, suckers. Okay. There are many types of diffusions. Okay? Many types. How many? There's osmosis, there's okay, there are two types of diffusion. Simple diffusion. Okay, by the way, this diffusion is going off the AP Bio textbook, and then facilitated. But what is diffusion? Diffusion is a sharing of things, or the spreading of things, right? Let's explain what diffusion is. I want to do this in the perspective of a water glass, right? Diffusion truly, truly means the movement of particles from a high to low concentration. How can I explain this as easy as possible? Let's say you have a glass beaker, okay? This beaker is filled to the brim with water. Right? And then, oh shit. It's filled to the brim with water. Right? And I drop a few drops of food coloring. Right? Now, if you guys want to understand how this works, you need to imagine that when you put food coloring in a glass of water, all that food coloring almost feels concentrated in that area. And then slowly, sooner or later, it spreads out. And soon, the entire beaker gets covered with that food coloring. That's diffusion. Those particles were in high concentration together. And then the movement of those particles spread out, making the water change color. That's exactly what diffusion is. Spreading something from high concentration, such as the small concentration of the food coloring, to a low concentration, to the water being changing color because all the particles are equally spread out. We could even think of this as a ball rolling down a hill, right? As we get a ball on the top of the hill, that's when it has the most potential energy. It's the highest point where it can release energy. And as it goes down, it lowers its potential and increases its kinetic. But we're talking about potential here. And as it lowers to the potential, the concentration of potential energy goes from high to low. And the same concept is here. The concentration of water are being spread out equally, if you say, right? Equally, right? So what is simple diffusion? What is facilitated diffusion? The easiest one right now would be simple diffusion, because I literally just gave you an example of it, right? Let's say we were at this a plasma membrane, right? Right? Let's say we're at a plasma membrane, right? I'm just gonna draw this as a plasma membrane, right? Actually, you know what? Work hard, not smart. Right? Let's say this is a plasma membrane. This is outside and high. This is inside and low. So this is the outside of the cell, and this is the inside of the cell, right? Now, let's say that there were a lot of these molecules. I'm not going to say the name of the molecules, just call them the Z molecules. Z, Heroic, whatever you want to name the molecules, right? These are just some molecules. It's a very high concentration. There's a lot of these molecules, but nothing on this side. So what's diffusion? Now, let's assume, okay? Let's assume that these all molecules are membrane permeable. That means they can easily go through the membrane. Just assume that for me, guys, please. Right? So, what does this mean? Is that these things, as I showed with the example of the watercolor, is that they need to spread out. They're in a concentrated area. The movement of the particles will require them to spread out. They have too much energy and they need to be spread out. Right? They need to be spread out. So, what do? Simple. This facilitated diffusion, the simple diffusion, just allows molecules to go here. And as you go, some of these get depleted because they're going to this side, right? And sooner or later, there's going to be an equilibrium, right? 5 here, 5 here. That's simple diffusion. It just passes through the membrane and easily makes it equal, right? It makes it into a low concentration, diffused evenly, which is considered as equilibrium. If it's diffused easily, it's called equilibrium. So that's a concept of diffusion, right? That's simple diffusion. It just goes through. Now, what's facilitated diffusion? Let's use the same concept here, right? Outside high, inside low, right? We'll just draw some more. Now, let's say that these guys cannot be semi-permeable. That means it cannot just easily go to the membrane. How is it going to go through? Because this energy should not be here. Plasma diffusion, high concentration of a particle where it can be diffused is not good. It causes too much energy to be there, and it can cause entropy. But we're not going to go into that detail. All we would need to know is that it does not want this concentration here. We do not want this. We hate this concentration, right? We want this to move, but they cannot move through the plasma membrane. So what do? Something called facilitated diffusion. To put into easier terms, let's consider facilitated diffusion. Facilitated diffusion is basically just a protein, really, right? Because it can't go through that fatty inner sides, because it's probably water-loving, what biology allows is these proteins to stick in between these plasma membranes, and they go fully through the plasma membrane, right? They go fully through the plasma membrane, and it's just this protein that allows the molecules to go in and pass through to be diffused. That's facilitated diffusion, where the particles get help by the protein that there is, right? That protein allows for the solute to be diffused, right? Okay, remember what solute is? Solute is particle, right? Solvent is a water. And then solution's a mix. Okay, remember that. But the protein, all it does is allows that diffusion to happen. Because if there was not that protein, then that means none of the actual particles can go through, and it won't ever cause that diffusion to happen. That's what facilitated diffusion is. The protein helps the particles to do diffusion. Really. Okay, if you guys want more detail on that, tell me. I will definitely help you on that. Okay. This is you can also call these, instead of simple and facilitated, you can call these passive and then active diffusion. right? What does that mean? What does that mean, right? What that means is passive. Passive as in, oh, I'm just chilling, I'm relaxing. That's what simple diffusion is. It just passes through the plasma membrane. And then active, it means actual work. It means that running. It needs that protein to do the work for them. And these proteins, by the way, these proteins require ATP to even work, to allow themselves to open up, to let those certain particles in, right? They require ATP. That makes them active. That's why it's called active diffusion and then passive diffusion. Right. And then concentration gradient. Concentration gradient, to put in layman's terms, right? Concentration gradient. Right? It's the the unequal distribution of particles, right? So to say in layman's term, wait, I'm gonna do AP because I have a really good definition that I saved. Right. Concentration gradient is when it's the gradient of diffusion, right? So concentration gradient looks something like this, right? Where it's something where there's a whole bunch, and then it slowly just becomes a lot less, then a lot more less, till there's nothing at all. Right? It goes from something that's super concentrated, highly concentrated, to something that's very low concentrated. Right? That's what it is. That's what it is. That's what concentration gradient is. It's a gradient of concentration. It's a gradient of the amount of particles in a certain area as it's lowering down. You know how when I said diffusion, right? How that glass beaker gets filled. It gets spread out with that solute, with that food coloring, and then ultimately turns into the color of the food coloring. It goes through a concentration gradient where it goes from very concentrated in one area to spreading out around the water to fully being spread out in the water. That's what concentration gradient is. All right. Okay, this is something good. Plant cells versus animal cells. Ooh, shucks. Okay. Wait, I'm gonna get a drink of water real quick, guys, because your boy is thirsty. ASMR. Okay, the next thing is plant cells versus animal cells. Okay, we just need to know the distinguishment between organelles. Now, there is a few organelles where they're not the same. Now, I'm going to pull up an image because I am way too lazy to draw them, and I do not have the time. I have to focus. The one thing that I'm really scared for is explaining the steps of mitosis and meiosis, because that's going to take a while. Right? Let me show you plant and animal cell. Alright, let's see what there is. Alright, this is a pretty good image. Oh yeah, some kids were asking what app I use. I use Colanote. That's the app called. It's really fun. It's also free, so don't worry about the pricing. It's completely free, so you have Colanote right there. Alright, this is the general sense. Okay, first things first. Actually, I do not like this image. It doesn't seem as detailed as I thought it was. Hold up, let me pull up a new one. You know, I try to give you the best of the best, right? I can't let these gummy things slide. Your boy needs to give you the best or else I'm not teaching you correctly. Mind you, you guys can always call me and text me and email me during the day of homeroom, right? You guys can please ask me questions. I don't want you guys to suffer. Okay. So there are a few things, right? Some things that they share, right? They share... they share mitochondria. They share nucleus, right? But what they don't share, that's what I like to talk about. Cell wall. Cell wall is a protective barrier around the cell membrane. That's what gives rigidity to the cell. That's what lets the cell stand up on its own, right? Animal cells don't have that. We're kind of flexible kind of guys. You know what I'm trying to say? Vacuoles hold liquids. Right? They hold liquids, they hold food, they hold nutrients. We have multiple of them throughout our cells. They have one massive one which holds a lot of water, which lets them to like increase in size, make them a little big, right? Makes the cell big, which allows them to have that kind of rigid shape. These two help in rigidity, and this one is a food storage during like winter months when they need the nutrients and they don't have viable access to them. Now the biggest one. Can you tell me which organelle is the biggest one where they have a difference of? You're right, beta. It is the chloroplast. This thing is so important for the cell. It causes photosynthesis. This is not in the animal cell at all because we get food from other organisms. They create their own food, so they use a chloroplast. Okay, so those are the major differences. Looking at it, I don't think there's any other differences mainly here, right? They have a cell membrane, they have microtubules, they have all that good stuff. Oh, wait, they don't have a few of the microtubules and centrioles. Like, when it comes to the cell transport chain, the cell transporting organelles, right? A few of them are missing, but she's not going to go into details about them. Central vacuole, cell wall, chloroplast, you know, that's basically it. Okay, our next topic is the characteristics of the prokaryotic cell. Let's pull up a whoa, this is gummy. Alright. And you know, big naked, beautiful, big T women don't fall off the sky, you know. I'm not high, you know. I've been searching high and low. Alright. This is a pretty good example. Can I like bring it? Okay, it's not allowing me to bring this guy. L. Yo, do not tell me I have to draw this. I'm not in the mood to draw. Your stupid friend is not. I'm not in the mood to draw, Ginny. Alright, perfect. Alright. This is the image of the prokaryotic cell. This is bacteria. This is archaea, right? Bacteria and archaea are listen. Bacteria and archaea are prokaryotic cells. Bacteria, archaea. They're both prokaryotic cells. Okay. I don't know why they did this. Okay. Let's look at the distinguishing factors. Distinguishing factors. Grr. First of all, no membrane-bound DNA. So kick out mitochondria, Golgi, all that thing. Anything with the membrane, anything that looks complex, no. Right? You see how there's these complex things? Yeah, it's not found here. Right? But what's the next thing? The next thing is the DNA. There is no nucleus to hold the DNA. Instead, the DNA is just chilling around the cell. And that's our circular DNA right here. The DNA is just there. It's just circular and it's just spinning around. Right? And there's a whole bunch of ribosomes. Those are helping in the DNA translation to make it into proteins. They have that. Right? The next thing is the flagella. This is a motor part of the cell. This helps the bacteria move. Think of it as like a tail that propels. Think of a boat motor, right? The motor of a boat, right? That helps it propel the boat across the lake. That's the same thing. It propels a bacteria around, right? The next thing is a capsule. It's a capsule shape, right? The shape is specific for the cell, for the bacteria, for the cell. Yeah. They have a cell wall, right? It has a cell wall just like a plant cell. Not as complex, but still a cell wall. And it's a capsule shape, right? They have silica all around them. Even the animal cells have silica, but these ones specifically are famous for silica, like paramecium, right? These silica also helps with moving around. They like how do I say? It's like caterpillar around. I don't know how to say. Like shake around. They just move with the hairs. That's so weird. But they do that. That's everything related to the prokaryotic cell. If there's anything else, please let me know. Functions of organelles. Oh, shucks. I hate this so much. Okay, this is the last topic for this video. And then we're going to move on to the next video where I'll finish the entirety of it. Actually, no. I'll finish like 4 topics. Then I'm going to do mitosis and meiosis. Ocean acidification. Yeah, it's going to be 2 more videos. Then I'm done. Right? Also, to those wondering when the video is gonna be pulling, the VC live chat coming pulling up, it's like 8:30 to 9 o'clock. So stay up late. Tell your mama, tell your papa, right? I'm not in the mood to do them any earlier. Okay, so let's focus on the three organelles. Functions of organelles. You guys should know this by now, right? You should know the main functions of every single organelle. I'm going to go through these ones, and then I'm going to go to the one that she specifically focused on. Ribosomes makes proteins. I'm going to write each of these down. Make protein. Lysosome. To put into perspective, think of them the garbage bin, right? Garbage bag, right? They take degradation. So they're the garbage bag. They take in waste. This holds genetic info. The nucleus holds genetic info. Nucleolus. What does the nucleolus do? Shucks. Sala. It's a center area. Center area of nucleus. Holds concentrated DNA. Concentrated DNA. That's where the concentrated DNA is. Mitochondria. Powerhouse of cell. Takes in food, creates energy, creates ATP. Rough endoplasmic reticulum. We're going to skip that and we're going to go into detail. Actually, you know what? Let's go into detail now, right? Rough ER, right? Embedded with ribosomes. They're embedded with ribosomes. This allows for protein creation where they then export it, right? Rough ER function, right? What it does is it does synthesis, the folding and the modification of the protein, right? The synthesis, the folding, the modification of the protein, where it then is in the vesicle and then it's like left, right? They create the protein with the ribosomes, embedded with the ribosomes, they make the protein. They synthesize it. That's the creation of the protein. They fold it, right? And they change it up of the proteins to make, secondary, tertiary, quaternary, all that stuff, right? They do all that things to the protein, make the protein itself, and then they transport it through using vesicles, right? Smooth endoplasmic reticulum makes lipids. They also make lipids. They detox, right? That's why your liver has a lot of smooth ER because they detox a lot of alcohol, right? And these both transport. That's very vital, right? That's the main functions of each of them. Vacuole holds water and nutrients. Peroximomes, right? Peroximomes, peroximomes, okay, function. I'm looking for a kid definition. Okay, their function is they break things down. That neutralizes diseases, right? Neutralize foreign invaders. And is just like the peroxide, they take out trash. They have acidic. Oh, they also are part of apoptosis. That or lysosomes. One of them kills the cells. Apoptosis. Okay, yeah. It's the which organelle. It's the lysosomes. The lysosomes do the apoptosis. They do cell death. Where's the lysosomes? Cell death. Oh yeah, lysosomes are also part of cell death, right? That's the peroximomes. Golgi apparatus. Think of them as a mailman, right? Take in and leaves or gives packages, aka vesicles, vesicles, modifies them to be able to ship, right? So remember those vesicles with all the proteins from the rough ear and the smooth ear? The Golgi takes it in, he does magic, he changes it up, and then he lets them leave. They go all around the cell, they leave the cell, they do something with them, right? That's not my job. Cell membrane lets things in and out of the cell. Cytoplasm. All right, cytoplasm function. There's a lot of functions. There's a lot of functions of the cytoplasm. It just houses and maintains organelles. Think of that. House and maintains organelles. Does some cellular processes. Does some basic cellular processes. Processes like glycolysis or fermentation is done in the cytoplasm. That's in cellular respiration. She didn't ask us to go in that, so I'm not going to worry about that. Okay. Oh, one last thing. Organic molecules. Organic molecules have carbon and hydrogen. Okay? Okay, sorry if you hear the vacuum. My bad. Do organic one. Sorry, guys, hear the vacuum. Listen, they have carbon and hydrogen, right? They hold both. They can't just have one. They need both, right? Lipids. Phospholipids are organic molecules. Water, surprisingly, is an organic molecule, but that's kind of given, right? I think Ms. Suarez might have written just carbon. If that's the case, then so be it. But I personally believe it's carbon and hydrogen. Check your notes if she says it's only carbon. Follow what Ms. Suarez says. Don't follow what I say. Remember that. I'm second best. Okay? If she said carbon, then it might be carbon. But I believe it's both carbon and hydrogen. So that's everything for this video, guys. This is part 2 done. There's like 2 more videos, and then we're done with this thing, right? We have 2 more videos, and then we're done. It's some few topics. The next video will be from defining isomers to steps of mitosis to meiosis. And then the last video will probably be like 5 minutes where I define oxygen acidification and explain central dogma in each step. This is everything you need to know for the bio benchmark part 2.